But at that time, we also proposed one way to do this, and that is, if you can somehow optimize your photocopying so that you work on a single DNA molecule, and assuming that you can do this counting on many of those DNA molecules, then we calculate that if that's the case, you should be able to arbitrarily determine the precision of assay. It's almost like I'm trying to do a survey of you. If I interview 10 people, I can only differentiate with a resolution of one person. But if I interview 100, 1,000, then I can actually even do higher and higher precision. So we calculate at that time that if I want to detect a fetus whose DNA has Down syndrome and which is present 5%, I need to interrogate over 100,000 single DNA molecules. So when we published that in 2007, many people thought that we're crazy. It seems that we are proposing that you need to do 100,000 tests per case to give you the answer. And one of those tests could cost you, say, $100, and then times 100,000, very expensive test. But interestingly, in 2008, with the increased popularity of a new type of DNA sequencing called massively parallel sequencing or next generation sequencing, suddenly we can actually interrogate millions or billions of those DNA molecules. Those are DNA molecules on a glass slide, and on one piece of glass slide, you can have actually up to 1 billion or 2 billion of those. So what we propose the following year is this. So imagine if you extract some DNA from mother's plasma, and then use those new DNA sequencer to sequence millions of DNA molecules. And because I know the human genome sequence, I can use a computer to map each of those molecule sequence back to the chromosome from which it comes from. And then I can plot a histogram showing the representation, the proportion of different chromosomes in the blood. For every chromosome, I can calculate what percentage of the sequence read is from that chromosome. And then you can have histogram like that, so chromosome 1 to 22, X and Y. And then you can see from the result, the longer chromosome have more reads, and then the shorter one have less reads. And then an interesting fit is that we then test this on a mother carrying a female baby, another one carrying a male baby, and then some adult male plasma, like from myself. And the most interesting thing is over there, when you can see, for example, if you are a man, I can see your Y chromosome in your plasma. But if you're a woman carrying baby boy, I can also sequence the Y chromosome from the plasma. If it's a woman carrying baby girl, I see nothing. But the most amazing thing is that on the X chromosome, the level of X in a man is approximately half of that woman. But the most amazing thing is that if it is a woman carrying a baby boy, the level of X is lower than that of a woman carrying baby girl. Because here, the mother is XX, the baby is XY, and whereas here, the mother and baby both XX. So XX plus XY is less than XX plus XX. So when I saw that data, I know we have cracked the problem. Because here, it shows you by sequencing the plasma of the mother, I can tell how many chromosome X the baby has got. If I can do that for chromosome X, I should be able to do that for any chromosome, even chromosome 21. So then we move very quickly to do chromosome 21 using the same system. And we decide to actually use some very simple statistics, something called Z-score, which is basically you measure the percentage in a particular case, and then you compare with a group of controls. And then you ask how many standard deviation is your case compared with control. So if there's no difference, then the Z-score will be around 0. On the other hand, if the baby has got more chromosome 21, then the Z-score will shoot up. And we decided to use 3 standard deviation as a cutoff. And interestingly, when I do that experiment, the result was very promising. So you can see, for example, the cases in green and red, the Z-score all very high. Actually, those cases, the baby have Down syndrome. And Down syndrome also called trisomy 21 because there's 3 copies of chromosome 21. On the other hand, the normal cases, those in orange and blue, the Z-score actually fluctuate around 0. So with this very early test, we only test 28 cases. Half of those have Down syndrome, half are normal. And we are able to detect it with 100% accuracy. So we're very excited with that. And then we decided to actually go and confirm the data with a large cohort of samples. So what we did is we collaborate with 8 hospitals in Hong Kong, 1 in London, and 1 in Amsterdam. So in total, we analyzed 753 cases, in which 86 of them have Down syndrome. And this is actually quite a major effort because a Down syndrome case is 1 every 700 pregnancy has Down syndrome. So imagine to get 86, you literally have to screen thousands, tens of thousands of cases. And in the end, this is the result. We're able to actually detect every single one of those Down syndrome cases. So the diagnostic sensitivity is 100%. For the cases which is normal, we're able to have an accuracy of 98%. So this work was published in January 2011. And since the publication, there's been numerous other studies confirming the result. Actually, the latest was there's a number of New England Journal articles about 2 weeks ago. So in total, now in the literature, there's probably over 20 or 30,000 cases in this area. And because it's so robust, that actually very surprisingly, 10 months after we published that first paper, we have already been able to license this out. And actually, our licensee was launched the test in the US in October 2011. And we introduced this to Hong Kong in December in the same year. So now, just over nearly 4 years after we launched the test, over 1 million pregnant women have already done the test, and it's available in over 60 countries. And it's actually one of the fastest growing genomic tests which has been launched. And also as a result of this test, the number of amniocentesis done in USA has actually dropped by 80%, which is quite incredible. And actually, this is paralleled by increase in non-invasive prenatal tests, which now people call this NIPT, non-invasive prenatal testing. So basically, after quite a number of years of work, we're able to actually finally have this development, which can make non-invasive prenatal Down syndrome testing a reality. But then, just as an academic exercise, I asked my student, and some of those are in the audience, like Irina, I said, okay, now we took us 10 years to solve this problem. Can you think of another way we can crack the same issue? So you can imagine if you were my student, you become very worried because you're worried